and you are free to start whenever you're ready.
All right, thanks, Jessa. So my name...
I don't see the screen, I'm afraid. I don't see the screen. I lost it for some reason. I have the evaluation, but I
can still see your screen, Alyssa.
Okay, it's still sharing?
Yes.
Okay.
Just click on the Zoom button.
A lot of time it disappears up into the upper right of your screen and you got to click on the image up there.
Okay.
Just move your mouse up there, it'll become.
Okay.
Or minimize everything else on your screen and that'll be the only thing left standing.
Okay. Yes, I got it now. Thank you. Right, I'm back.
Okay. So my name is Alyssa Grove, and I did my thesis on current and power production in two-chamber high-temperature microbial fuel cells with Dr. Bruce Logan as my thesis advisor working in his Logan lab. So the reason that we are interested in microbial fuel cells as an area of research is because the current energy needs require the burning of fossil fuels, which release greenhouse gas emissions that are the main driver of climate change. The United Nations Environment Program has recommended cutting emissions drastically in order to mitigate the worst effects of climate change. So the solution to this is to explore and develop alternate clean sources of energy so that we can decrease the impact of the environment that we currently have with the most common ways that we get our energy currently, while also meeting the energy needs of a growing population, particularly energy for industry, transportation, and particularly wastewater treatment. Because as I'm sure we're all aware, freshwater is extremely important. So wastewater treatment uses 3% of energy and produces up to 2% of emissions. And so reducing these emissions and demands could assist global endeavors to drastically cut emissions. So one solution to this problem is using bioelectrochemical systems, or BESs, to utilize the chemical and thermal energy contained in wastewater, as this substance contains more energy potential than what is currently used to treat it for our manufacturer and consumer energy needs. So in short, wastewater could become a net energy producer instead of a net energy consumer. A popular type of BES is microbial fuel cells. They basically work by having two electrodes. One is the anode, as you can see on the left here, and the other is the cathode, as you can see on the right. The anode is negatively charged, the cathode.
Excuse me, Alyssa, I'm only seeing slide number 1.
Do
you have it in presentation mode?
Yeah, it's in presentation mode.
Is anybody else seeing more?
It's not in presentation mode.
Thank you.
Hang on. I think I know the problem. Okay, can you see it now?
Yes.
That's much better.
Thank you.
Okay.
Thank you.
Sorry, Zoom. I thought that Zoom would follow the window when I put it into presentation mode. Thanks for speaking up. So,
okay.
yeah. So this was the introduction slide that I was seeing, but nobody else was. So, so here's the diagram of microbial fuel cells, and
no,
basically
there we
how
go.
They work.
Now we're seeing it. Okay.
Yeah.
Yep, we can see it.
I think it's showing the, yeah. So the anode is negatively charged and the cathode is positively charged, so that, and they're connected by a resistor, so that we can track current and voltage and then power using Ohm's law. So the microbes essentially collect, the microbes that are typically used in MSCs are called exoelectrogenic microbes, which means that they have the ability to transport electrons outside of themselves and onto an electron acceptor, which I will explain more about in a minute. But basically, the microbes will deposit electrons on the anode, and then these electrons will be attracted through the wire and resistor towards the positively charged cathode. At the same time, protons are generated at the anode, which then migrate across an ion exchange membrane depicted by the vertical orange line in the middle of this diagram towards the cathode to rejoin with the electrons that have been transported over to the cathode. This maintains the negative charge on the cathode, maintains the positive charge on the cathode so that it won't become negatively charged over time, and then that will kind of stop the attraction of electrons towards the cathode. And what I was talking about with electron acceptors and electron acceptors and electron donors, basically it's kind of analogous to what humans do. So what we do is we eat sugar, which is essentially carbon, and we collect the electrons from that carbon and we exhale carbon dioxide. So we use oxygen as a final electron acceptor. The same thing is happening in the anode chamber of the microbial fuel cell, but they just use different sources of what we would use carbon for, and then they also use different final electron acceptors other than oxygen. So exoelectrogenic microbes typically transfer electrons in 3 different ways. In this diagram on the left, it depicts the exoelectrogens using chemical mediators. In the middle, it depicts conductive pili that the microbes grow over time so that they can funnel the electrons across the pili to the electron acceptor. In this case, that would be the anode, and then through direct contact by just connecting to the anode and directly depositing electrons. So the exoelectrogenic microbes that I used were hyperthermophiles, which basically means that they grow at very high temperatures, as you can see here, between roughly 70 to a little bit over 100 degrees Celsius. And we were really interested in using that because in theory, the hypothesis was that hyperthermophilic temperatures are less susceptible to microbial contamination. So hyperthermophilic microbes would potentially would compete less with other microbes because the microbes that they would be competing with can't grow at such high temperatures. So they would essentially die as they became too hot to survive. Whereas other microbial fuel cells are run at mesophilic temperatures and are more susceptible to contamination. And in addition, chemical reactions occur faster at higher temperatures. So that was another point of interest because they can be used to generate chemicals or produce more current because of the higher temperature, creating a more favorable chemical reaction. The first microbe hyperthermophile that I used is Geoglobus ahangari. Researchers found this in hydrothermal vents in California at a depth of 2,000 meters. It grows between 65 and 90 degrees Celsius, but its optimal growth temperature is 80 degrees Celsius. It's an obligate anaerobe, which means that it can't tolerate oxygen at any level, and it uses hydrogen and iron-3 as hydrogen as an electron donor and iron-3 as an electron acceptor, which provides it sufficient energy to grow without the presence of organic carbon. These researchers also discovered that this hyperthermophile uses short and long chain fatty acids as electron donors for iron-3 reduction. So this faster growth, and it also grows faster on long chain fatty acids. And this is possibly due to the hyperthermophilic temperatures of its environment. And it also has environmental significance, as microbes with this ability may play an important role in breaking down organic matter in hot microbial environments. And previous researchers working with the Logan lab have showed that it produced a current when grown under optimal conditions in a microbial electrolysis cell. The other hyperthermophile that I used for my experiments is from shallow hydrothermal vents in Italy. It's also an obligate anaerobe, and it uses iron 2 and hydrogen as electron donors and nitrate and thiosulfate as electron acceptors. So it grows between 65 and 95 degrees Celsius and with an 85 degrees Celsius optimal temperature for growth. Previous researchers at the Logan lab also showed that this microbe produced a current grown under optimal conditions in a microbial electrolysis cell. So first, for microbial fuel cell assembly and inoculation, we first assembled microbial fuel cells using aseptic techniques and pure culture techniques, which basically means we sterilized every component and the growth media, the analyte and the catholyte that we used in the MFC. And so first we would make the media, and we would either autoclave or filter sterilize the separate media components before we would use them in the microbial fuel cell chambers. For the proton exchange membrane, which you can't really see it in this photograph, but it's clamped between the joint connector of the MFC depicted here. So basically in the middle of the clamp. We were using Nafion, and we needed to first sterilize it before we used it in assembling our MFC. So first we would boil it in hydrogen peroxide and then hydrate it in sterile distilled water. Then the graphite brush anodes inside the reactor chambers here, inside the glass bottles. We baked them in a muffle furnace to remove all potential contaminants. And then the separate components, the clamp, the 2 reactor bottles, and the caps, we autoclaved those separately, wrapped in tinfoil, and we assembled these MFCs inside of a, we assembled them inside of a, slipping my mind, inside of a chamber to maintain sterile conditions, and replace the analyte and catholyte using pressure feeding as depicted here. So basically what happens is we would force anaerobic gas into the analyte and catholyte chambers. That would increase the pressure, and that would force it through the needle and tube leading to these waste collection bottles on either side, so that we could empty the bottles anaerobically, because we were concerned about opening them and just dumping it out, because that would expose them to oxygen, and we didn't want to risk that. And then we would have bottles of fresh media ready before we started doing this, and we would increase the pressure in the media bottles to force it into the anode and cathode chambers to replace the fresh media as the microbes grew and used up the resources that we provided for them with the old media. For open culture techniques and non-sterile techniques, we did largely the same procedure, except we did not autoclave the bottles, clamps, or reactor chambers, and we didn't boil the Nafion membrane in hydrogen peroxide. But I did bake the graphite brush anodes in a muffle furnace to remove potential contaminants before using them in the MFCs. We then inoculated for both aseptic and open culture microbial fuel cells. We inoculated them 3 each for Geoglobus ahangari and Ferroglobus placidus, and we incubated the ones inoculated with Geoglobus ahangari at 80 degrees Celsius and the ones inoculated with Ferroglobus placidus at 85 degrees Celsius. We then connected them to We then hooked them up to a resistor box at the bottom here. And a resistor box, basically, you can essentially choose what resistance you use it at. So those little switches that you can see on these resistor boxes, that's the resistance level that you can set it at. So we set ours at 1000 ohms. The resistor boxes are connected to the MFC, as you can see at the top with these wire clamps. That's how it's connected to the resistor box. And then they're also connected to a Keithley multimeter, which is then connected to a computer to monitor current production. So after we did that, it was eventually determined that the media itself was producing a current, and at comparable levels to inoculated MFCs. So we decided to discontinue these initial MFC experiments and design an experiment analogous to an MFC reactor. So that's what we did. So the idea with using serum bottles as opposed to MFCs is that I could then simulate the contamination procedure that we had been planning to do on the MFCs themselves. And so what I decided to do was use to take wastewater and serum bottles and inoculate serum bottles with the wastewater after it was filtered to remove large particles. And I would inoculate serum bottles to be incubated at room temperature, so around 20 degrees Celsius, at 80 degrees, and then incubate another set of serum bottles at 80 degrees Celsius, and then another set at 85 degrees Celsius. And then I would monitor the number of live cells within each serum bottle. I also inoculated serum bottles using pure culture techniques with Geoglobus ahangari and Ferroglobus placidus to measure the number of live cells that they had. And the reason that the inoculums were kept separate is because I would be monitoring the number of live cells using microscopy techniques. And under microscopy, you can't really tell different species of microbes apart. So if the live cells were kept separate, then it would be more easy to determine which type of cells were alive and which type were dying at certain temperatures. So the way that I monitored the number of live cells is through live dead cell counts. And the way that this works is you have 2 different types of fluorescent dye, and one shows up green under a certain wavelength of light, and the other shows up red at a different wavelength of light. So 2 different microscope filters, essentially. And so then the green dye dyes all of the cells in a sample, whether or not they're alive or dead. And the red cells dye only the cells that have died by dyeing only cells that have broken membranes. But the red dye can't penetrate an intact membrane. The green dye can. And the theory is that cells with broken membranes are dead because they're compromised, essentially. And that's how you can tell the number of live cells from the number of dead cells. And you basically count these up under a microscope. And then you average out the number of live cells, and then average out the number of all cells and dead cells, and then you have, then you calculate the total number of cells and the total number of dead cells. And then you take the difference of this number, and that's how you can tell the number of live cells. So using these calculations, I was able to monitor the number of live cells within a cell sample and over time. So this is a comparison of the serum bottles inoculated with wastewater growth over time. And I also did this for Geoglobus ahangari and Ferroglobus placidus, but they are not depicted here. But because when I started to monitor the number of live cells with the Hyperthermophile species, Penn State had ordered the labs to close for undergraduate research, so I did not have that resource anymore. But they did, the Hyperthermophile species were shown to be increasing steadily over time. So the wastewater cells on the left is the number of live cells at 20 degrees Celsius room temperature. The middle is live cells at 80 degrees Celsius, and the right is live cells at 85 degrees Celsius. So starting on the left with the room temperature, it showed that cells were decreasing slowly over time at room temperature. And this is because the wastewater that I was using is an anaerobic wastewater kept at 40 degrees Celsius at a wastewater treatment facility. So at room temperature, cells are dormant and might die off slowly or are growing very slowly, because for thermophilic or hyperthermophilic species, room temperature and lower is essentially like being frozen for them. So they are dormant where mesophiles tend to grow. In the middle, the live cells over for the serum bottles incubated at 80 degrees Celsius also decreased steadily over time until they reached zero cells. And the one point here showing an increase towards the end, I think, was an error when I was counting out the cells. And one of the bottles, I think it's bottle one. Bottle one shows an increase towards the beginning, and I think that this is because as the serum bottles heated up, they reached the temperature that the microbes were accustomed to at the wastewater treatment facility before the wastewater was collected for experimental purposes. So they became active again for a period of time before the serum bottles became too hot for them to survive and they started to die off. And then the last sample set on the right, the 85 degrees Celsius, bottle one again shows an increase and then a decrease. Because for the same reason, I think that they must have reached temperature so that the microbes became active before dying off because it became too hot for them to survive. So the results are that the populations of the hyperthermophile species, Geoglobus ahangari incubated at 80 degrees Celsius and Ferroglobus placidus incubated at 85 degrees Celsius, they increased steadily over time for serum bottles prepared using both open culture and aseptic techniques. Populations of microbes within the wastewater that was used to inoculate serum bottles at room temperature 80 degrees Celsius and 85 degrees Celsius. For the room temperature ones, just decreased steadily over time or held roughly steady because of the dormancy of the microbes, potentially. Or at the hyperthermophilic temperatures, they first increased and then decreased until no live cells were left. For some concluding statements, wastewater contaminated serum bottles incubated at high temperatures showed no live cells after reaching oven temperature for some time. So what this could mean is that high temperature MFCs fed with ambient temperature wastewaters don't need to be assembled and maintained using aseptic techniques. And this could impact the researcher community by meaning that researchers could save time and money by not having to ensure sterility of parts, tools, and growth media. Because sterilizing techniques can take a lot of time and energy with the use of autoclaves and muffle furnaces. Future experiments. Similar experiments could be performed on serum bottles, but they should be repeated on MFCs to supplement the results obtained from what I did with these serum bottles. And also temperature should be monitored as well, in order to also supplement what I was theorizing about the growth media reaching temperature and sort of activating the microbes and then becoming too hot, which resulted in the microbes starting to die. These experiments should also be repeated multiple times on MFCs and monitored for longer periods of time than what I was able to do at this time due to the ongoing COVID-19 pandemic. And then another area of experimentation and research that could be done is also to determine what component of the media was generating current. I would also like to thank the Logan Labs for their support of my thesis project, as well as the Rada Erickson Discovery Grant for funding my thesis research in the summer of 2019. These are just some references, and if anyone has any questions.
So, okay, let's see. Let me ask you a question. And I don't think I would have thought of this question 3-4 months ago. But if microbial fuel cells were widely used, could they become the weapons in the hands of terrorists? How will they impact society? How could they impact society negatively?
I'm not sure if they could be, because let me go back to my assembly and inoculation.
Mm-hmm.
Essentially, the MFCs, it was a lot of work to assemble and maintain them, as well as particularly when I was using the aseptic culture techniques to assemble them and maintain them, because of all the initial steps to ensure the sterility of everything, as well as inoculating them, because the hyperthermophiles that we used, they came from, we tried to inoculate serum bottles at first to make sure that the microbes were first alive that we were using. And the first ones that we inoculated were with hyperthermophile samples taken from a negative 80 degrees Celsius refrigerator, and they were dead. And then we tried a 4 degrees Celsius refrigerator, and those were alive because they were growing, we could see them. So right there is one area of difficulty, is obtaining microbes essentially that are alive and viable for inoculation. Secondly is the difficulty of sterilizing everything to make sure that the microbe that you want is the one that you're growing. And then third is if terrorists wanted to use like MFCs, are you saying that terrorists would want to use them to inoculate potentially dangerous microbes to humans? Because those would need to be more at mesophilic temperatures or like living human temperatures, which are more susceptible to contamination. And also, the microbial fuel cells could be used to generate gases such as methane, but you have to collect it and go through other processes with it in order to make it usable, essentially, because it's methane gas, not like ethanol or something. So I don't think it's a very viable option for that particular.
Well, the reason I asked is because I read today in the morning that this coronavirus, the presently interesting coronavirus, has been found in several locations in Paris, in wherever they treat waste.
Mm.
That might be because of so it might be because people might be like coughing or like spitting into a sink, and then that is transported to the wastewater treatment facility as a result, because it gets rinsed away with everything else. But coronavirus is a virus, not a microbe, so it would have to infect a microbe that is an exoelectrogenic microbe, but then that would kill off that because viruses take over a microbe, essentially. So something like a virus wouldn't really, I don't think it would work, because it doesn't have the same properties as a microbe in terms of like how they live and their life cycle and things like that.
Okay.
If I may, actually, Professor Laktaki kind of stole my question. So I'm going to rephrase a little bit, but before I do that, just one possible, just minor grammatical correction. You kept referring to microscopy. I think it's usually pronounced microscopy.
Oh.
Unless it's something different. I mean, that's a minor thing, but anyway, I wasn't necessarily thinking in terms of terrorism, but one of the things I was just wondering about this method is that there's no such thing as a free lunch. You pay a price for everything that you do. So if you're extracting energy, so obviously you're burning fossil fuels, you're generating carbon and all sorts of other particulates that create health problems. What's the downside to these types of fuel cells?
Right now, the downside is that they produce small currents instead of the level of current that would be needed to make it a viable consumer source, because it's more in research and development stages to make it a consumer, like a viable consumer product, essentially. So that's kind of the downside, as well as the current, like the also efforts to scale them up are facing issues with also scaling up the resistances and other issues that are not as prominent at small levels, like so for small reactors that are typically used in labs. So there's difficulty in scaling up to large, like to making them a bigger component that would be required for typical wastewater treatment facilities with the amount of wastewater that they need to treat every day, essentially.
Okay, that's not what, I mean, those are engineering problems, all right, that eventually you might be able to get past. What I mean is when I say the downside, not that, well, it's not as efficient or productive as we want it to be, what's the price that the environment pays for it, or what, I mean, I don't know that much about them, but I just, everything else, like, you're burning fossil fuels, you're releasing carbon, and what what else might be in there that you're burning. So this, using biological processes, but are you, obviously it's bacteria, it's not viruses, so you're not releasing new viruses and creating new pandemics, but there has to be some price that you're paying for this energy that's detrimental, or I'm assuming, maybe it's not, maybe it's the greatest thing since sliced bread, but you know, what can you envision any downside to it, that it's emitting? I don't know. I'll leave it.
So some MFCs use methanogens, so they produce methane, which is a greenhouse gas. So that could be a downside as just what the microbes do. Essentially, so that's something that could be a downside. In terms of the net energy producer, the reason, going back to the introduction, one of the reasons is that these MFCs, that's the wrong slide, these MFCs are being investigated is because wastewater contains more energy potential than what we currently output to treat the wastewater. So that theory is that it would be a net energy producer. But with depending on what microbe species you use, so if you use like a methanogen, you could be producing methane, so you're right back at square one.
Okay, yeah, that's perfect. And actually, my understanding is that pure methane is more of a greenhouse gas than if you burn it, or, you know, the byproducts of combustion. So that's exactly what I was looking for. Good, thank you.
Yeah, that's a growing problem with the like raising cattle, essentially. They're trying to cut methane emissions from the cattle.
Oh yeah.
I could chime in on that too, Alyssa. I think you did a good job of pointing out the main issues. I think the issue is that we currently use lots of microbes. That picture you have there of the wastewater treatment plant is a big microbial farm, right? It's like a biotechnology plant, and you're right. We're growing all sorts of microbes there, and if we were to grow pathogenic ones in the microbial fuel cell, that would be a real problem. But in fact, there's very little evidence to show that the ones that grow in this way are pathogenic. So that's not, from what we know, an issue in terms of pathogenicity. And then, like any other treatment plant, after it's treated, you chlorinate it to disinfect that wastewater, and then you dechlorinate it to get the chlorine out so you don't release that. So there's some checks and balances that occur too, but the potential for turning CO2 into methane as a side product in the process is one that would be potentially detrimental, and I think you pointed that out pretty well.
Thanks.
Yeah, Alicia, I got the impression that the work is intensive, good work. There's a lot of work involved.
Yeah,
And,
worth it though.
Yeah, I'm struck by that, by the amount of work involved. So the objective is to optimize the energy generation for this particular bacteria, right? That's the overall objective, right?
Yeah, and other species of microbes with the same electron transport properties as well.
Well, I wonder if the membrane has an interest, a role that's of interest.
Yeah, so what the membrane does is, the typical membranes that are used is a proton exchange membrane. So it selectively allows protons to migrate from the anode chamber to the cathode chamber to help maintain the positive charge on the cathode. Okay. And without allowing media crossover.
Very nice. And then I just have a question about the global impact of this. Are there nations where they're using biogenerations and waste management coupled with waste management activities?
Sorry, could you repeat that? It cut out at the first half of your question.
I'm wondering if there are globally, if there are nations or companies that utilize this energy generation coupled with waste mitigations. Are there examples around the globe where these kind of technologies are being employed? Biogeneration and bio?
I don't think they're being employed yet. It's more in the research stage, essentially, to develop the technology to the point where it could be employed, and have more energy potential to be used, essentially. But I think this is research being done, from what I remember reading about. It's being done in, I believe, China and in the US, from what I remember.
That's interesting. Thank you. Good work.
Thanks.
Okay, I don't know what happens from here because I haven't done one of these before. So,
I was wondering if anybody had any more questions.
yeah. So, I mean, we go until the questions are over, and then is that it, or do we do anything else after that?
I think that's it. Other than you guys filling out the rubric.
Okay.
I think.
Yeah, you guys can take her out for an ice cream, however.
Yeah. Yeah, I'm gonna go ask my mom for lunch.
So what's your plan after this?
After
After graduation?
Applying for jobs. I don't know how that's gonna work, but applying for jobs.
Well, why don't you go for a PhD in sustainability? I think that's
Yeah.
a very important topic. And
Yeah, so I think that getting some work experience before going for a PhD would be good for me in terms of getting more work experience. But also,
I would disagree with that, but unless you have, and I'm being very, unless you have, debts because of being a student, go on directly. It's difficult to come back.
Mhm.
Well, anyhow, good luck.
thank you.
See you in class in a couple of hours.
Yep, thanks.
All right.
Good luck, young lady.
Thank you.
Bye-bye.
Bye.
Good job.
Thanks.
Excellent.
Yeah, good job, Alyssa. I think nice presentation and nice summary of the field. You obviously have done your homework and learned a lot in your time here. So I was glad to see it, and sorry about the COVID stopping everything short there.
Yeah, thanks.
Everybody at this meeting probably has regrets about that because we're having this over Zoom. So say no
Yeah,
more.
yeah. I'm taking a BME class this semester, and the teacher kind of explained to us that it's like essential research or long-term projects only. So basically medicine, or if you have a really long-term project going. So
The senior thesis didn't quite sneak in under that bar.
yeah.
All right. Well, very good though.
Thank you.
I think that's it then, unless anybody else has any other thoughts.
I'm good.
Okay, great. Thanks, Alyssa.
Thanks for coming, everybody.
Okay, bye.